Welcome to the Brainweaver Project. Today we will learn more about these fascinating creatures and the safety measures required to handle them. Let's get started. The parasite creature known as a brainweaver has a body no larger than a or a couple of grains of sand. The initial encounter with a brainweaver is deceptive, as it appears as an innocuous speck of dust, easily overlooked by the naked eye. Its lifespan without a host is only about 24 hours. It gains entry into the human body through the inhalation of contaminated air or by making contact with an open wound. Once inside, it embarks on a perilous journey toward the brain, using a network of microscopic tendrils to navigate the intricate pathways of the nervous system. Upon reaching the brain, the brainweaver deploys specialized enzymes and toxins that allow it to penetrate the protective barriers of the brain tissue. Once inside, the parasite taps into the neural circuits, skillfully intertwining its tendrils with the host's neurons. The brainweaver possesses a unique ability to release a potent blend of chemicals, causing the host's brain to undergo a gradual transformation. It releases a combination of neurostimulants and neurotransmitters that enhance the host's susceptibility to its influence. These chemicals weaken the host's cognitive and emotional faculties, making them increasingly pliable to the parasite's will. Over time, the brainweaver establishes complete control over the host's brain, manipulating their thoughts, emotions, and actions. The once independent individual becomes a mere vessel for the parasite's intentions of spreading its species, completely unaware of their own subjugation. This process can take from 6 months to 3 years. As the infestation progresses, the brainweaver exploits its newfound control to further its survival. It compels the host to seek out and infect others, perpetuating its spread. The infected individuals become carriers, unwittingly aiding in the parasite's expansion as the brainweaver manipulates their behavior to create ideal conditions for transmission. As you can establish, these creatures are not to be taken lightly, but the potential this organism has to offer is too great to not take the risk. The last 2 years of experiments have given us invaluable insight into this creature. Not all organisms are compatible. In our tests, we found that only a third of all mammals are compatible. The exact reasons for this incompatibility are chemical and also to a degree psychological, it seems. A more relaxed and susceptible state of mind of the organism improves the chances of infection. About a quarter of humans are found to be compatible. In the cases where humans have been incompatible, a physical change occurs. Slow or rapid, the body will begin to deform as the parasite is desperately trying to stay alive in a body that is not accurately responding. Most of this deformation appears in the face and head until too much for the human to stay alive, killing also the brainweaver. Handling brainweavers requires utmost caution due to their parasitic and potentially dangerous nature. Here are some essential safety precautions that should always be followed. Containment measures. Brainweavers should be handled within a designated containment facility, such as a Biosafety Level 3, BSL-3, or higher, equipped with appropriate engineering controls to prevent accidental release. This includes controlled ventilation systems, secure doors, and restricted access. Personal protective equipment, PPE. Laboratory personnel must wear appropriate PPE, including gloves, lab coats, protective eyewear, and respiratory protection if necessary. The PPE should be worn at all times when working with brainweavers to minimize the risk of exposure. Training and education. Laboratory staff should receive thorough training on handling and containment procedures specific to brainweavers. They should be knowledgeable about the parasite's characteristics, transmission routes, and proper handling techniques to ensure their safety and prevent accidental contamination. Engineering controls. The laboratory should have adequate physical barriers and equipment in place to prevent accidental exposure. This may include specialized containment hoods, sealed containers, and secure waste disposal systems. Strict aseptic techniques. All procedures involving brainweavers should be conducted using strict aseptic techniques to minimize the risk of contamination. This includes sterilizing equipment using sterile techniques when handling the parasite and regularly decontaminating the laboratory surfaces. Waste disposal. All waste generated during the handling and study of brainweavers should be considered biohazardous and disposed of according to the laboratory's standard operating procedures for handling infectious materials. It should be properly labeled, securely contained, and incinerated.